Hello everyone, welcome to another video. This is going to be the first gram-negative bacilli video, which is the Enterobacteriaceae. So the Enterobacteriaceae is gram-negative, and they are known commonly as the enterics. They are commensals of the human as well as the animal intestinal tract, where they form fecal coliforms. They are ubiquitous, meaning they are found almost everywhere. Some of them are pure commensals, meaning they don't cause any disease, whereas others have some pathogenic factors. So the characteristics is that they are aerobic, but they are also facultative anaerobic, meaning they can live in an environment without oxygen. They can live but not thrive. They are easily cultured. They reduce nitrate, ferment glucose, and is catalase positive. Some of these Enterobacteriaceae are capsulated. Those with the biggest virulence factors are normally capsulated. Okay, so transmission can occur in two ways, exogenous and endogenous, meaning that exogenous is when the organism or the microorganism is transmitted from an outside source, such as contaminated food or water, like E. coli in dams and rivers contaminating drinking water and food. And then endogenous is from the host's own intestinal flora. They are mostly opportunistic infections, but they can also occur through medical or surgical interventions. Then classification. So the primary pathogens, these are the ones that are capable of causing disease. I have an abbreviation for this. It's KEYS. So it's Klebsiella pneumonia, and then it's E. coli. Both of them can be primary pathogens as well as opportunistic pathogens. So it's Klebsiella pneumonia, E. coli, Yersinia, then it's Salmonella and Shigella. So these five are the primary pathogens of gram-negative bacilli Enterobacteriaceae. And then the opportunistic pathogens, these cause a disease under certain conditions. There are Proteus, Enterobacteriaceae, and then also E. coli and Klebsiella. Then lactose fermenters, they are E. coli or Escherichia coli, Klebsiella subspecies, and Enterobacter. Whereas and the non-lactose fermenters, there are Salmonella, Shigella, and Proteus. Then motility, there are also E. coli, Enterobacter, and Salmonella. The non-motile is Shigella and Klebsiella. Okay, then pathogenicity. So once again, there are primary intestinal pathogens and there are opportunistic intestinal pathogens. So these primary pathogens are true pathogens and are normally not part of the the normal flora in the gastrointestinal tract. And examples of them include Shigella and Salmonella. And then you get opportunistic pathogens. These are normally commensals of the gastrointestinal tract, and they cause infections in susceptible hosts. Okay, so let's look at the virulence factors. So a capsule is always a big virulence factor. They are responsible for immune evasion, evasion of phagocytosis, and things like that. And examples are E. coli, Klebsiella, Salmonella. And then the liposaccharide, the lipid A, the oligosaccharide core or the O antigen. They produce endotoxins, which protects the organism against the complement system. Lipid A stimulates macrophages to produce cytokines. Okay, and then the next virulence factor is urease. This is a very important virulence factor because this potent enzyme will hydrolyze urea to ammonia and carbon monoxide. And this urease enzyme can be the main cause of the formation of kidney stones. So it will hydrolyze urea to ammonia and carbon monoxide. Then fimbriae or pili, this is normally for adhesion. So it allows for the bacterial adhesion to mammalian cell surfaces. You get different types with the different receptors. Um, and then toxins. So you get endotoxins and exotoxins. Um, Exotoxins are normally associated with gram-positive bacteria, whereas endotoxins normally are associated with gram-negative bacteria. And these toxins facilitate three things: the adhesion, which is adhesion to the host cell, which is type 1, damage to the host cell, which is type 2, and evasion of the host cell, which is type 3. Um, Examples of this are examples of some pathogenic E. coli, which is enterotoxigenic E. coli. They produce two toxins, LT and ST toxins, and this will stimulate fluid secretion, so diarrhea. And then the next virulence factor is genetic exchange. So normally any bacteria has this, which you can gain DNA rapidly from organisms through mobile genetic elements such as transposons, phages, or plasmids. Normally most bacteria has plasmids, and this plasmids mostly will cause antibiotic resistance. Then laboratory identification. So of course it's a gram-negative bacteria, so the bacteria will appear pink. It's bacilli, so it's rod-shaped, and they are aerobes but also facultative anaerobes. And then also you get lactose and non-lactose fermenters. The lactose fermenters were E. coli, Enterobacter, and Klebsiella, if I remember correctly. Where was this? Yeah, so the lactose fermenters were E. coli, Klebsiella, and Enterobacter, whereas the non-lactose fermenters were Salmonella, Shigella, and Proteus. And then there's also some of the clinical syndromes that can be associated with Enterobacteriaceae, but we'll do this further in the further blocks. And yeah, this is the lecture on gram-negative bacteria, Enterobacteriaceae. Thank you for listening, and like and subscribe.